I'm Marianna Bacellar-Galdino. I'm part of the Department of Ophthalmology at Scranton, Pennsylvania, Altasciences. We work in preclinical ophthalmology, and I'm here to present to you a poster about a geographic atrophy-like induced model in nanopigs. Geographic atrophy is one of the signs of age-related macular degeneration, or AMD, and it refers to the retinal pigment epithelium, the RPE, cell death that precedes the photoreceptor death, which will then lead to blindness. The nanopig is an ideal model due to the eye similarities to the human eye. So the pig retina has broad photoreceptors dominant periphery and cone photoreceptor dominant visual strip. The objective of this study was to develop a large animal model for geographic atrophy in nanopigs injecting sodium iodide subretinally. We used 3 nanopigs that received bilateral subretinal injection of sodium iodide, which created this bleb that you can see on the RETCAN image on day 1. So we could also see the damage on the fluorescing of the machine, and we could follow up this bleb in the OCT sections as well. The layers of the retina were also measured as total retinal thickness, inner and outer segments of the retina separately. So all 6 eyes received a successful 100 microliters injection of sodium iodide subretinally, with the blebs being able to be observed on all time points during ophthalmic exams and fundus imaging. The retina was reattached on day 3, as you can see on the OCT image on the poster on day 3. And the FA, the fluorescing showed increased signal at day 3, and then later a little less on day 8 and day 14. But the damage could still be seen as the marked bleb in the retina. There was a significant decrease in the total thickness of the retina in the inner retina and in the outer retina in the bleb area, proving that the sodium iodide created the damage. The total retinal thickness decreased by 53%. The inner retina in the bleb area was decreased by 28%, and the outer retina in the bleb area lost 74% of its thickness. The retinal thickness outside of the bleb area showed no changes in any of the layers, and also no changes in the total retinal thickness by day 14. And inflammatory-like cells were observed in vitreous and in the retina during the OCTs. With this, we concluded that the subretinal injection of the sodium iodide created a successful geographic atrophy-like lesion in nanopigs. The significant thinning of the outer retina indicates a loss of photoreceptors similar with what is seen on the AMD disease, and this localized retinal damage mode will allow us to screen and the development of new therapies and drug candidates for dry AMD. Thank you very much.